So I want to take you back to April 2023. That's when, you may remember, Professor Al Oy from the U of R had a presentation here of his research on the origin of species, which took him to the Solomon Islands. Shortly thereafter, he became the biology chair at the University of Rochester and started the Trop Bio Lab. Trop is short for tropical. Most recently, his research work was highlighted in the 2024 full issue of the U of R Review magazine. And last month, if you were here or online, you remember Maria Castagna, also a member of the Trop Bio Lab, met with us to discuss her research in Colombia. And now we have the third member of that lab, and possibly the last one that I'll get. So let me introduce to you Dr. Elsie Shogren to speak to us. She grew up exploring muddy creeks and observing wildlife around the family farm in Nebraska. She channeled her fascination with biology as an undergraduate at Cornell University, where she discovered field work in ornithology, collecting data on tree swallows in Ithaca and Alberta, Canada. After graduating, Elsie continued developing her research interests, assisting with projects on the Red-backed Fairywrens in Australia, greater Prairie Chickens in Nebraska, and Piping Plovers in South Dakota. Deciding to focus on the interplay of sexual and natural selection for her PhD at Kansas State University, she studied the consequences of rainfall for survival, behavior, and evolution of sexually selected Neotropical Manakins. And now, as a postdoctoral researcher at the University of Rochester, Elsie is now exploring the genetic ecological, and behavioral factors that maintain species boundaries between two Myzomela? I missed it. Yep. Honeyeaters that have recently came into contact and are hybridizing in the Solomon Islands of the South Pacific. Please welcome Dr. Shogren.
Thank you so much for the very kind introduction and invitation to come and chat with you all. I'm very excited to tell you a little bit about a recent project that has sort of come out of some data that we've been collecting over the past few years. And before I dive into the science, though, I did want to sort of just expand a little bit and maybe show some cute bird pictures to give you an insight into sort of how I came to be in the room talking with you this evening. So as David mentioned, I grew up in Nebraska, which if you haven't been, does frequently look like this. I had a lot of fun growing up on a farm, mucking around, riding horses, chasing my dog around. And I was also really fortunate to grow up in a family that was aware of birds and valued going out and watching them. So I got to go and see the crane migration through the Central Flyway in the Platte River of Nebraska. Is absolutely a life-changing event. So if you haven't gone, I'm sure many of you have made it, but I would highly recommend it. And so I knew that I wanted to study biology. I knew that I wanted to try to spend some time outside. I went to Cornell University. I figured they probably had some different options that I could explore. And there I discovered the Cornell Lab of Ornithology. But while I was there, I was actually working with fish. So my first job as an undergrad was working as an assistant in the Cornell University Museum of Vertebrates on their fish collection. So I was working with organizing and cataloging and adding to the collection of fish at the CUMV, which they have a really nice collection there. But eventually, in that environment, I discovered birds. I took ornithology my sophomore year, and it very much changed the trajectory of my career. But it also sort of was a natural extension of my family. And sort of an interesting anecdote is my grandmother, Marjorie Crimmings, also went to Cornell for undergrad. She was a badass. And she really enjoyed birds, and she passed on that love of birds and nature and learning about the world to myself and my two cousins, Alice and Sophie. We're all within a year of each other, and we all wound up going to Cornell together. And I had this really surreal experience. Senior year, I was a teaching assistant in ornithology. And so it was the morning of class. I was going through the trays, finding the teaching specimens that we were going to put out for the class to look at. And I flipped over this tag number and read Marjorie Crimmings, my grandmother's maiden name. And this was a specimen that my grandmother had prepared 60 years prior while she was an undergrad at Cornell. And it was just this really surreal, incredible experience. This was about a year before she passed, so I was able to call her on the way to class later that day and be like, Graham, I found this bird that you prepared, and it's still in the teaching collection, and we're using it to share our knowledge and our love of birds. And so it was really special. We were able to all take a picture of it with my Graham's bird. And so I discovered birds. I was pretty set on birds. And I also discovered, in addition to sort of learning about their evolutionary biology, I really also enjoyed the field work. And so I got my start as an assistant working with the tree swallow project in Ithaca and in Canada. And this was, just daily going out, monitoring chick development and growth. And I just loved it, and I wanted to figure out how to do more of it. And so after undergrad, I wasn't really sure what questions were going to motivate me. So I decided that I needed to get a little more exposure, build on that skill set. And so I started off going to Australia to work on the red-backed Fairywren with Daniel Baldassari, who at that time was a grad student and is now a professor just down the road at SUNY Oswego. So if you need another talk person in the future, I highly recommend. He does really cool stuff. So I was in Australia for about 4 months, and then I came back quite a bit closer to home, worked on greater prairie chickens, looking at their lekking behavior, their movement, their nesting ecology, and association with wind farms in the sandhills of Nebraska. And so that was sort of a really cool opportunity to just like sit and watch birds doing what birds do when they're being absolutely ridiculous on their booming grounds. And then my final tech job, as I was gaining experience, was working with the piping plover population. There's a central US population of piping plovers that nests along the Missouri River, and Virginia Tech has been doing a lot of really great work with them for many years. And so this was a lot of just surveying, cataloging territories, looking at survival, that kind of thing, and getting to catch really, really adorable little baby fluff balls. And so at this point, I had sort of narrowed in what it was that really motivated me and made me fascinated about birds. So I decided I was ready for grad school. I went just a little bit south to Kansas State University, which is in these really lovely Flint Hills of eastern Kansas. It's a beautiful place to live and work, but I couldn't resist the call of the tropics. And so I wound up working with Neotropical Manakins as a PhD student at Kansas State. And so I was really interested in how natural selection and sexual selection interact with each other, and particularly how rainfall in these really rainy environments might limit these birds' ability to dance or display on their display areas. And so I did some comparative work with different species, but I primarily focused on this guy. This is the white-ruffed manakin, Corapipo altera, and I studied them in Costa Rica. They live in these really lovely mid-elevation primary forests. And manakins do their displays. They're one of these species where the males are doing displays in designated areas for females. Females come and check them out. In this species, the display area is this fallen mossy log, and they're quite fastidious about their fallen logs, and they clean it. And a lot of times if you're walking around and you're trying to see if you're finding a display log, you'll look and there's little peck marks in all the leaves right around the display log because it's not quite right, and he's trying to like tidy it up for a potential mate. And so anytime I found one, I would do a lot of behavioral observations. So that's a blind. It's a highly technical conglomeration of PVC pipe and black plastic and, and some shading material to try to stay as cryptic as possible. And so I really can't help myself. So I'm going to show a video of some manakins displaying just because. So there's a female. She's very cryptic and green. She's on one of those mossy logs. And that's the male doing his big flat, we call it a flap chihuahua display. And he goes up way above the canopy and he dives down and he makes this noise, flap chihuahua. And it happens so fast that we decided we needed to take high-speed video cameras in there and see what was actually happening. So we've got a high-speed video of that happening. And so you can see there's this backflip, and then they do this really slow, stylized, we call it butterfly flight. And he's just floating away on a cloud, and it's delightful. So that was my PhD, was getting to watch all of them. I had a wonderful time. But eventually that came to a close, and I was really fortunate to have an opportunity to get an NSF postdoctoral fellowship to come work with Al at the Trop Biolab at the University of Rochester. And this has been an absolutely incredible experience. I've learned so much from Al, from other members of the department, being a part of this really dynamic, very exciting place, gotten once-in-a-lifetime experiences of going to the field with Maria in Colombia and seeing her cool birds. And I also did get some teaching experience last fall. I was able to go teach a study abroad class in the Galapagos for some U of R students. So that has all been absolutely fantastic and has sort of prepared me for the next chapter. And so you all are getting the absolute last talk that I will give as a postdoc at the University of Rochester, because Sunday I'm moving to North Carolina. So I am starting a lab at Wake Forest University in January. And so if you pull up the Wake Forest Department of Biology, you can see the Shogren lab is going to be right there. So that was just sort of a little bit of background in terms of how I've gotten here. And so now we're going to dive into the science and talking about some pretty cool little birds called honeyeaters. Okay, so before I start, I need to make sure to acknowledge that a lot of times I might say I did this, but really what I mean is we did this, because this is very much a collaborative project. And Al has been working in the Solomon Islands for 20 years now, and so I am a relative newcomer to the team. We have an incredible team of local collaborators. Lonsdale, James, and George have been working with Al for that 20 years, and so they really have an incredible collective knowledge and experience that is invaluable to our efforts. Nisus, Johnson, and Mage are sort of a younger generation that have started more recently, but these guys are absolutely integral to everything that we do, and so it's very much a collaborative project. I get to talk to you about it today, but I just want to make sure that you realize that this is really the work of a collaboration between ourselves and our local counterparts in the Solomon Islands. So we all love looking outside at all of our birds. This is one of my favorite artists, Charlie Harper, and he, is representing the biodiversity of Monteverde in Costa Rica. And it's just such an incredible array of motmots and quetzals and trogons and manakins. And one of the questions that comes to me, at least, as we look at this incredible assortment of organisms is, how are they all coexisting? How are they all occupying the same space and able to all survive? And this is a question that's been sort of with our field since Darwin, is how do organisms share the same space? And we frequently have an ecological perspective where we think about they're limiting their similarity, or they're partitioning different resources. So, MacArthur's foundational work with the warblers sort of led us to see that, if you're limiting how much you are overlapping, you might be able to coexist stably because you're reducing competition between each other for food in this context. But there's also an evolutionary perspective. So you might be occupying the same space, but if you're all interbreeding with each other, then that's going to make it pretty difficult for you to remain distinct taxa. And so in this case, we might expect to see assortative mating, where different types are mating with a similar type in order to sort of keep those evolutionarily distinct trajectories where these organisms can be in the same space, but they are in fact different species. But of course, we know that both ecology and the evolutionary aspects are interrelated. But disentangling them is frequently complex, because you might have ecological factors which drive evolutionary change, but then those evolutionary changes are going to change the ecological dynamics. And so it might be helpful, if we want to try to understand this interplay, to look at systems in which we have taxa that are closely related. They aren't too different from each other. And they've come recently into contact with each other. They're sort of still figuring out those dynamics. If we roll up and it's been 2 million years, they've already figured out how they're going to partition everything and how it's going to play out. So if we can come into a system where there might still be some of that give and take going on, we might have an opportunity. And so Meyer, in his sort of foundational work on allopatric speciation, came up with this model. If we think about how species evolve, He proposed that you have a single population represented by this one blob. And then if it becomes separated, geographic isolation, so down here we have two different blobs, then there's an opportunity for differences to arise. So you might have different color patterns or different ecological niches coming about. But if those two closely related taxa then come back together, this is the point at which we can see, okay, have they differentiated enough that they can coexist and occupy the same space? Or are those differences either from an ecological or an evolutionary perspective not strong enough, and so they would just merge back and collapse back into a single, so they're not really coexisting, they become back into one species once again. And so Meyer with Diamond in their book about the birds of northern Melanesia called this the moment of truth for speciation, but I think it also provides a really key way to ask about how species find a way to coexist. And so if we look at this moment of truth, what is it that's allowing these closely related species to occupy the same space but remain distinct, identifiable species? So today we're going to be talking about the honeyeaters, the family Melophagidae. There's just over 50 genera, 195 species. Most of them feed on pollen and nectar. There are a few that have adaptations for and feed on insects as well. And there's just a remarkable array of colors and shapes and sizes, and they're found all throughout Australia and Oceania. We're going to zoom in particularly on the Solomon Island honeyeaters, so that's right about there. We're going to zoom in there, and we're going to focus on honeyeaters in the genus Myzomela. And so if we look at the Myzomela honeyeaters in the Solomon Islands, there's usually one species per group. So the red-vested Myzomela in Malaita, the crimson-rumped Myzomela, Guadalcanal has the black-headed Myzomela. But there's one island group down here in the far southeast corner where the story changes a little bit. So on the Makira Island group, we have the big island of Makira, the smaller satellite islands of Ugi, Three Sisters. On the big island of Makira and then Santa Ana and Santa Catalina, we have this all black sooty honeyeater, Myzomela tristrami. And then on these islands to the north, Ugi and Three Sisters, we have a red cardinal Myzomela, Myzomela cardinalis. And so this is an instance where we have two species of Myzomela quite close to each other. And in 20th century expeditions, some folks started cataloging what birds were where. So focusing just on the big island of Makira. In 1908, A.S. Meek was going out and collecting for the Rothschild collection. In 1927, Ernst Mayr went with the Whitney South Seas expedition that was funded by the American Museum of Natural History. They got to work off of this really cool boat. That's Mayr right there. It looked like it was fun, different kind of field work. And then in 1953, Kane and Galbraith went with an Oxford University expedition. So we have some really interesting historical perspectives on what birds were on the island of Makira. And so we're going to look at that data first. So prior to 1908, we only saw blackbirds on that big island of Makira. This is the native endemic species. Then in 1908, Meek found a bird that was mostly black but had some red feathers. And so I'm going to show any of these sort of mixed appearance birds or phenotypic hybrids in sort of a blue throughout the talk. So we had our first record of a hybrid. And then Mayr actually in 27 was the one who first recorded pure red birds on the island of Makira. And then since then, in 1953, there was this huge peak. They found a ton of red birds on Makira. And then 21st century, we've gone back between 2008 and 2015, and then most recently in 2022. And it sort of seems like they're settling in around 50-50 of both the red and the black being present on Makira. So we now can update our map to show that the red species is in sympatry, is in the same place as the black species, but it seems like that region where they are interacting with each other is quite restricted to the coastline. So if you roll up to a beach in any of these places where we've got the red and gray hash mark, if you're right down at the coast, you'll probably see redbirds and maybe a few blackbirds. But if you go further inland, you're going to start finding only the blackbirds. So it really seems like they are restricted in how far they can invade. However, they are very similar in their size and their ecology and the behavior. So if we just do a side-by-side, they're both around 11 to 16 grams. The reds, the males, can be a tiny bit bigger, but really very similar. They both feed on nectar. They do this really cool behavior where they're holding on with their feet hanging upside down and then sticking their bill into the flower. They're socially monogamous. They have an open cup nest. On average, they have 2 eggs. Every once in a while, you'll find just a single egg clutch. And so given this similarity, it's not that surprising that they would interbreed with each other. And so this is sort of the range of hybrid individuals that we have caught over the years. And it ranges everywhere from, maybe just a few more black feathers than you really expect there to be in a red bird, to almost completely dark birds with just a few very subtle red feathers. And often they get them on their throats. They kind of look like hummingbirds. It's really, really remarkable. So we do see evidence of interbreeding, but you'll notice if you go back to this graph that there's still relatively few that have that mixed appearance. So it seems like they are still keeping a little bit separated. So we wanted to know, how are they coexisting? And we have a few different options for how that could be. This is non-exhaustive and certainly not exclusive. But one way that they could be doing is that partitioning of space, that the reds and the blacks are living in different parts of the island. The other option is that they could be in the same space, but they could be using different resources. Going back to the MacArthur example with the warblers, maybe they're using different trees as their food resource. And then the third option, going back to more of that evolutionary perspective, perhaps there are differences in how they choose mates or what happens when they interbreed. Maybe they're mating assortatively, that is like with like. And so that's something that's keeping them distinct as red and black. And they're not just all interbreeding willy-nilly. So we'll start off looking at whether or not we can detect if they're partitioning the space that they're in. And so this involves capturing birds, and again, incredible work from our local team of setting up mist nets, climbing. These are canopy birds, and so we have to get nets pretty high if we want to have good success catching them. And so they do some pretty incredible stuff to winch nets up and climb up and make it possible so that we can set up nets. And if we're lucky, then we'll catch some birds, and we'll be able to catalog their identity, their phenotype, what they look like. So if we zoom in, so we've primarily, well, you can't see super well, but we were zooming in. This is the region of the island that we focus our work on. If you zoom in there, we're going to first look at the total number of individuals of each different class, so red, black, and then those mixed phenotypic hybrids. And if we look over the entire region that we have sampled, this is data from 2022, overall you see a slightly higher frequency of the red. So about 57% of the birds that we catch are red, 41% are black, and then about 2% are mixed. So right now it seems like there is a slightly higher frequency of reds, but we can then partition it into sort of an inland sampling versus the coastal sampling. And there things get a little bit more interesting. If we look at the inland sampling, it's much closer to 50-50. But if we look at the coast, it is much higher. We have 67% of those coastal birds are red compared to 29%. However, it isn't, if we compare that to the overall total, it's not significantly different. So we can't say that they're for sure partitioning. There's a trend in that direction. And then also important to note that this is at least at the scale of our sampling. So if we had gone further inland, perhaps that would have changed and we would have not seen hardly any red birds. But sort of dealing with the constraints of time and logistics and getting all of us and the gear inland, at this point, we don't seem to see them partitioning space if we're looking within a relatively small spatial scale. So then the next question is, within that spatial space that they are occupying, are they partitioning different resources? And so this was, again, our data from 2022. We got really lucky. We were basically the first people to get to the islands as soon as it opened up after COVID. And we were super fortunate because that coincided with a really great flowering event of this really striking pink flower. They call it an apple tree. It's definitely not an apple. I don't know what the scientific name is, and I should figure that out. But we were really fortunate because we were basically able to find, these trees that were flowering. You can see all of the pink petals on the floor or on the ground around this tree. many times this is right in the middle of a village. And so someone was like, oh, we have a flowering tree in our village. You should come and catch some birds. And so we would go and we would set up nets and we would, try to put nets up inside the tree. And so it's drawing in a lot of birds from the area, and we can see who we catch at each tree, but we're, sampling a single tree. And so if we zoom in on, just this is anecdotal, but a couple different days where we caught enough birds to be able to have some sense of, we've done a good job sampling on that particular day. Here at this coastal site, on the same day at the same tree, we caught 9 blackbirds and 11 redbirds. So they're definitely using the same resource. A different day further inland, again, we caught 12 blackbirds and 13 redbirds. So they do seem to be using this particular resource at similar rates. So it doesn't seem like that's particularly happening, at least in this instance. So then the third option that we can look at is whether or not they are mating assortatively. And so to do this, to try to understand the patterns of mate pairing, we need to find nests. And they're very small nests, and they're very high up in trees. And so we took this opportunity to really loop in a lot of community engagement to help us find nests. And so we did lots of presentations at community gatherings, chatting with folks, and the chief of various villages, and pounding the pavement, and getting out there, letting people know what we were doing, what our research was like, and what we were looking for. And this was all really made possible because Al has built these really long-standing, robust relationships with the community. And so, as I mentioned, he's been going there for 20 years. He has, an adopted set of parents, and it's really been an incredibly rewarding experience to kind of get looped in on that journey, because it really feels like a homecoming. We spend a lot of time chatting with, local government, national government, being really embedded in the community. And this sort of makes these kinds of projects possible because we have this buy-in, and it's a relationship between them. I've done a lot of outreach, having high school students come out into the field with us so I can show them what we're doing, and allowing them to sort of look at the bird, really close. It's you see the birds flying around, but it's just so incredible when you look at the bird right there. You're like, oh my gosh, it's so tiny and delicate. And then Al has really built on this more formally in working in partnership with communities to put together conservation efforts. And so this is something that's featured in the Rochester Review article, if you get a chance to read it, where he's really been working with them to establish this Yato conservation area, which is an incredible tract of 30,000 hectares of pristine forest that they're working to put under national protection so it can't be logged. And so it's been really very inspirational to be able to witness this kind of partnership with the local communities where we're collecting these data and being able to study these birds. So to get back to the mating assortatively, so once we would get a report of a nest, and we would go out and confirm it using binoculars, make sure we knew who the parents were at that nest. So there's some little babies right down there. So this is a very nice red nest. And then where possible, we would try, in addition to just identifying the color of both parents, we would also try to capture them. And so this again involves some gravity defying skills. And I don't recommend carrying your machete in your mouth, but ultimately we're able to put up nets through some pretty incredible skill sets to be able to capture the parents. And then once we have them, we take all kinds of measurements of their body size, their morphometrics. We take pictures for future reference so we know what they all look like. And I also put color bands on, so there's unique combinations of colors. So if we go back to the nest and we try to do an observation, we know who mom is and who dad is. And in future years, if we come back, we'll be able to revisit those areas and see if the birds are staying in the same area or not. And then we also take blood samples for genetic analyses. And then again, where possible and where it's feasible, and their idea of feasible is very different from my idea of feasible, but they do also, in some cases, we can get to the nest itself, and so we can take the chicks out very briefly to be able to put bands on them as well and take their genetic data. And so then we're working on a project where we can look at sort of these families and try to understand how the genetic information is being passed from one generation to the next. And so this is, yeah, really exciting work that there's a lot of different projects that are going to come out of this. But for today, I'm going to talk about the mate pairings themselves. And so overall, we had 2 field seasons in 23 and 24, where we were able to identify 69 nests. And this is sort of the area, again, where we're focusing our area in Kira. And so now we're going to look at the number of pairs on the y-axis, and then these different types of pairs. So homotypic, meaning the same type. So red, and black. And we see quite a few of the red, and the black pairs across that full data set. However, we do see heterotypic pairing. So these are a red with a black, or a hybrid with a black, or a hybrid, and we see all of the possible combinations. So they all have the potential to mate with each other. But we wanted to know what our expectation would be if they were just randomly pairing. And so to test this, we went back to that banding data that we had from 2022, where we had all the numbers of red and black and hybrid that we had. If we just randomly drew a pair of individuals out of that data set, what would our ratios look like? And so if we do that, if we just randomly take 2 birds out of our full hat of all of the birds that we know are in this population, what we see is a very different pattern, where we would have way more, over 50% of those crosses should be heterotypic, with red and black pairing with each other. And that is very much not what we see when we look at our observed data. So I think we can really conclusively say that these heterotypic crosses are happening much less commonly than we would expect them to. So they do happen, but they're not happening at the rates that they should be if there really isn't any assortative mating. So it seems like we can say that there are assortative matings because we see so much fewer of those heterotypic matings than we would expect if we were just randomly drawing individuals out of a hat. However, as we were sort of plotting this data and I was looking at it, all of these dots are the location of each nest. And I sort of was squinting at it, and I decided that we should draw a line from each nest to the closest point on the coastline. So that's what all of these little lines are, is just from that GPS point where the nest was, the closest distance to the coast. And if you plot that, we'll look at distance to coast along the x-axis, and then the number of nests on the y. You can kind of see there's a little bit of a bias, right? There's a lot more red nests happening closer to the coastline than those black nests. And we can quantify that. Again, this is the distance to the coastline along the x-axis, but now we're just looking at the number of mate pairs. There's more red nests closer to the coastline. The black pairs are further inland, farther from the coast. And then the hybrid nests kind of fall right in the middle. And very satisfyingly, this is statistically significant. So the red and the black are significantly different from each other in how close they are to the coast. And then the hybrids fall right in the middle and aren't significantly different from either of the parentals. So it seems, to me at least, that possibly this assortative mating that we're observing is being influenced by where birds are deciding to put their nests. And it seems like that nesting behavior is species specific. And so that could be contributing to who they mate with, is because of where they are mating. So overall, in terms of what it is that seems to be influencing how these red and black birds are coexisting, I don't think we quite know the answer for the partitioning space yet. We're planning to go back next summer and do some more transects farther inland, sort of plan for more extensive sampling, do transects maybe in at least one and maybe a couple of different places where we can really look at how the environment is changing and how the frequency of all these individuals are changing. For the partitioning resources, we don't have any evidence for that yet. Again, that might change if we have more data on different types of trees. We have sort of this one year when we had a really consistent flowering of one type of tree, and we were able to say, at least for that tree, there's no partitioning of resources. But then for the mating assortatively, I think we have some pretty strong evidence that there is something about who they choose to mate with that is influencing their the evolutionarily distinctness. And we also have genetic data that's informing this as well, and is sort of a different project. But this is really nice to be able to look at the behavior of the birds themselves and sort of corroborate what we're seeing in the genetic data with some firsthand observations. So despite similarities, it seems that these distinct species are sort of finding a way to coexist within the island of Mercure in this recent contact. And so it seems perhaps mate recognition or differences between the two species are preventing them from interbreeding. This might also be influenced by where they are deciding to nest. And in terms of, more specifically, what it is that's driving this red species staying so close to the coastline, we don't really know that yet, and that's another area of future work where we need to characterize the environments that these birds are in, both when on Mercure where they are coexisting, and also on Oogie and Three Sisters where they aren't interacting with each other, and so we can kind of get an idea of what the baseline is and look at what it is when they are in the same space, and see whether that has to do with some physiological or behavioral limitations. But overall, I hope that I have told you a little bit about the Solomon Islands and these pretty cool little birds. It's been an honor and a privilege to be able to do work in this corner of the world. So I'm really, really excited to be able to continue working on this, even though I'm moving to Wake Forest. The nice thing about academia is that you still have the opportunity to collaborate with folks. So I'm looking forward to continuing to build on this story as a professor at Wake Forest. And so one more shout out, funding sources, of course, the NSF supported me, the University of Rochester. The Trot Biolab has been an amazing place to be a postdoc, and our incredible team of local collaborators. I should say they're all wearing, so the bird in the indigenous language, Kikito, is the name for the Myzomela. And so they call themselves the Kikito Boys. And so when I went back last year, I was like, I made t-shirts for everyone. And they were pretty excited about it. So we had to take a group photo with everyone wearing their Kikito Boys t-shirt. And so with that, before I ride off into the South Pacific sunset, I would be happy to take any questions that you might have. Thank you.
Have you looked at the differences in the sex of each?
Yeah.
So are there more redbirds, males coming in, or
Oh, I
that
see.
type of thing? Have you
Yeah, so the question is for, do I see any differences in the sort of the invading species if there's more males coming into the population from Oogie, the smaller islands? Yeah, so I don't know for the initial invasion part, if there was a bias, there may have been. One thing that we do see in the genetic data is all of those hybrid crosses are the product of a red female and a black male. We can look at the sex chromosomes, and we see that all of the progeny of those crosses have, yeah. So that's what we see in the genetic data. However, when we went to look at this nest data, that was one of the big questions. We're like, okay, this is what the genetic data is telling us. There's only one cross one way. But we did find a couple of nests where the female was black and the male was red, or he was a hybrid. However, so that was exciting. But what we know is that there's a lot of birds that look black or look all red, but they actually are genetically mixed. So they're cryptic hybrids. And so we have to look at that black, potentially black female's genetic material to find out if she is herself a hybrid. And we have that data. We just extracted all of the DNA. I need to send it out for sequencing. So hopefully we'll get the answer on that. But it seems that at least, there may be differences genetically in their preferences that might be influencing that cross. The other possibility is that there are differences genetically where that interactions between genetic material doesn't work well in certain directions. So sometimes the cross of, a male of one species and a female of the other species works fine. But if you flip that and do the reciprocal cross where it's the other cross, then those alleles no longer interact well with each other. So we have to figure out if that's what's happening in this system. But yeah, no, it's a very, very good question. Working on it. Yeah.
Do you find any blackbirds moving over to Oogie, is the name of the island?
Yeah, very, very infrequently. They definitely haven't established a population there. There's sort of, if you ask folks, they're like, oh yeah, I think I saw one like 20 years ago. So I think it could happen intermittently if there's like a big cyclone or something, maybe they get blown off course. They're quite close. It's only like 8 kilometers to the, so you look at low tide and you're like, it's right there. Like, why aren't you flying over there? But it really seems that they haven't, maybe the competition is too intense on that island if all of the red species there is quite established. And so we're not really sure, what it was that brought the red species over and allowed them to establish, but it seems like it could have been multiple. There could have been failed attempts of invasion, and then eventually there was enough of a toehold that they started to expand. And the other thing is sort of thinking about the physics sort of it is, you're going from a really big island to some very small islands, and the other way it's like very small islands to a really big island. So if you get blown off course from Oogie or Three Sisters, you're going to hit Makura if you go south at all. So sort of the size of the target in terms of how these birds are moving is a little bit different, and I think might lead to a preference for the red ending up establishing on Makura rather than vice versa. Yeah.
And does it have anything to do with the numbers of, or lack of numbers of males on Oogie that forces the females over to Makura?
I don't know. I think those populations are pretty healthy. So I don't think there is any reason to think that there might be movement due to a lack of mates or anything like that. I think it's probably more of a random chance event that ended up rather than sort of a pushing out, as far as my understanding of it is. Mm-hmm. Yes.
Any other food sources other than the apple?
Mm-hmm.
Because apparently the apple tree seems to be happening with both groups. There could be possibly other food sources that are maybe more predominantly for the red.
Yeah.
Coastline.
Mm-hmm.
Versus going in a little
Mm-hmm.
bit.
Yeah, absolutely. And so, I mean, yeah, the black has evolved. It's been on the island of Makura for millions of years, right? That's it's evolved on that. So there could definitely be food sources that are there that the red species hasn't co-evolved with, isn't aware of. The other thing that we think about a lot is those coastal regions are quite disturbed. There's human habitation there. There's gardens. There's, they plant the flowering trees and things like that. Whereas you go further inland and it's more primary forest, then we think that the distribution of those resources might be more scattered. And so it just might be less easy to find those resources for the redbirds as they go further inland, whereas the blackbirds have evolved in this environment and are better adapted to it. But I think, we've sort of started this project from a genomic perspective, trying to understand genetic interactions, and we're sort of bringing it back to the natural history, and there's a lot of stuff that we don't know yet, including exactly what resources they're using. But part of this Yato conservation area is this really nice primary forest. There are rangers who are doing transects and sort of checking in on things and doing surveys, cataloging the biodiversity within this. And so we're hoping to set up a partnership with them where they can sort of do some of these surveys and record, what species they see the honeyeaters feeding on and try to get a better sense of what resources they are using in the interior. And then we can also do that kind of work both at the coastline where they're interacting. And then also, I think importantly, it will be good to go to the smaller islands where the red came from and try to compare, okay, so is the resource distribution and the species that are available on Oogie and Three Sisters, are those matching what we're seeing at the coastline of Makura? Is it sort of just a preference thing in that they are suited to this kind of habitat? So yes, those are all excellent future directions to try to get at some of those more ecological differences that could be influencing the interactions and how well they do in each environment. Mm-hmm.
I have a question and a comment from
Okay.
the Zoom audience. So first, a comment, Steve points out the apple tree. The proper name is the Malay apple, and forgive me, I'm going to butcher the scientific name. It's the Syzygium malacensi.
Oh, excellent. This is
Yeah.
great information.
And
Can he email me?
yeah, of course. And another question, are the hybrids fertile?
Are the hybrids fertile?
Mm.
That is a great question. So we know that some of them are because we do see individuals with varying amounts of genetic information. You know, some are predominantly red with a little bit of black genetic information. Some are more black with a little bit of red. So there have to be some fertile hybrids in order for that to be passed on, right? But the critical thing is how fertile are they? And are there differences in you know, maybe some combinations of the genetic information do work and they are able to find mates and raise offspring, but maybe some combinations aren't. Maybe the egg doesn't hatch. Maybe it hatches, but it's just a really not great you know, bird, and so it doesn't survive to find a mate and build a nest and all that good stuff. So I think there's probably variation depending upon the genetic makeup of the hybrid.
Thank you. And that's it from the Zoom audience.
Okay. Yes.
So, this is that the density around the island.
Mm-hmm.
Seems number one of the maps showing that all the way around.
Mm-hmm.
Or they concentrating where you saw them to begin with.
Yeah. You picked up on a very key nuance there. So the question was that the range of contact between the red and the black was sort of around I can't find it's fine, around the edge of the island, but it doesn't completely encircle the island. There's a region on the south side of the island where we still only see the black species. And we think that that region has expanded over time. So Al has been going there, as I said, for 2 decades. And so in 2022, we went around, we were able to expand that map. We went around to the other side and we recorded the red species sort of around the tip of that north part of the island. And there were no records of that prior to that. Yeah, there wasn't any information about that prior to 2022. So we think that they, between 2015 and 2022, had made that expansion. However, on the south end, that stop has been there for a long time. And we know that it's a hard stop because a lot of our team, our local collaborators actually live in that area of the island. So they know what they're looking for and they are paying attention. And they have told us that there's, once you turn that corner and get to the south coast, you don't see the red species anymore. And that south coast is called the weather coast because it's a little bit harsher. That's where the winds come up. It's a lot rainier. And so that is something that we are very interested in trying to figure out if there's something about the environment there. The black species does fine, the red species can't hack it. Because it really seems that even though it's started to expand to the north and around, it really hasn't made it the other direction. So that's one thing that I think would be really useful to be able to do these transects right across the island from the south side to the north side. And we're talking about putting up some weather stations so we can get year-round information about wind speeds and humidity and precipitation to try to characterize that aspect of how the environment might be related to where we find the birds. Yes, very, very good to notice that. So right, I see what you're saying. Yeah, so in terms of when they nest, you know, these past 2 field seasons were the first time we ever looked for nests. And so in 2022, we went and you can tell juvenile birds because they have yellow at the base of their bills if they've come out of the nest recently. And so we went in August and early September of 2022, and we caught a ton of juveniles all over the place. And we decided we wanted to go back and try to find nests. We're like, okay, so if we're catching juveniles in August, let's try going in June and see what we come up with. And we got super lucky the first year and had a ton of nests in June of both species. We went back again this year, this past summer in June, and it wasn't I think we missed the peak a little bit. I think it was an El Nino year, and so things might have been a little bit different. So I don't think we have enough data year to year. Yeah. But we do find both species nesting at the same time, within the month window that we're there, and we're, all hands are on deck trying to find nests for us. We do find them nesting, but yeah, there could be some more fine scale differences. And I think they're pretty, again, we have a couple of years of data, but it'll be interesting to go back in future years at slightly different times and see if we can figure out if they're nesting asynchronously or if there's a really dedicated breeding season, but we just don't know that quite yet. Yeah, definitely a good thing to keep adding to our information about their biology.
Thank you very much.
Oh, we have one more.
Oops.
Are red female by black male pairings as common as black female by red male pairings?
No. Nope, the black female pairings with hybrid or red males, I think of our 12 hybrid nests, I think only 2 of them were a phenotypically black female mating with a red male. The rest of them were red females with black males or with hybrid males.
Okay.
Yeah.
That's it.
Yep. So we'll see.
Thank you very much.
All right.
For your presentation.
Thank you all so much. Appreciate it.